All right. Thank you everyone for joining today, and thank you to the speakers. Welcome to the second part of the GoPHA webinar series on redesign for circularity. We're going to discuss some upstream solutions for applications recognized as critical. In the sense of regulatory priorities, also discussed in the treaty in a similar way. We have our speakers today, Dr. Kirk Havens, Dr. Yasmina, Dr. Martin van der Zee, and Ipsita Roy. And we'll hear from them soon. Some housekeeping rules for the webinar. Each speaker will present for 12 minutes, and we'll address all the questions after everyone has presented. But feel free to write your questions and direct them to specific speakers in the Q&A chat box. A little bit about GoPHA. GoPHA is a non-profit organization with 60+ stakeholders from academia, brands, material producers, everyone. It's a platform for everyone that is involved in driving a circular economy with the pollution-free alternatives for applications based on bio-based materials. And we operate on three pillars of policy and advocacy, community engagement and communication and education, and science and systems programs, all of which interact with each other to inform our work and priorities. We have been involved in the INC negotiations for the development of a global plastics treaty. Since its inception, we are an accredited UNEP NGO. And we present our recommendations. We facilitate information exchange, knowledge sharing directly with national delegations directly involved in these negotiations, as well as producing some knowledge products that are valuable for all sorts of stakeholders involved. An overview of the UN Plastics Treaty before we begin. Currently, all sorts of substitutes and alternatives, as they're broadly recognized, include non-plastic substitutes, bio-based, biodegradable materials in the treaty, and they're being explored due to their potential of reduced environmental impacts and also options of feasibility of scaling them up. We believe that the value or the potential of these materials to contribute to the goals of this treaty should not be underestimated. And negotiators must consider advancements in knowledge and make provisions to incentivize material use that is sustainable and circular in design. The significance of the treaty is such that it can drive a shift towards a defossilization of material use, develop a unified vision and mandate globally, and also present a roadmap of how to go about it. And a supportive treaty that focuses on upstream solutions like substitution with or use of better materials and not fossil-based plastics could lead to unified policies for scaling up, for accessing global markets, for accelerated research and development, and also for market growth for these materials. But we do face some challenges in terms of how the text could shape up and its interpretation, because there is lack of harmonized information or consensus of what is a suitable alternative and when it should be applied. There are some areas like, what about the chemicals and additives used for performance of alternatives and substitutes, globally accepted assessment criteria for these materials, and also the distinction or the treatment between conventional plastics and better materials, as we call them. In terms of assessing the need for better material use and design strategies, we will take this discussion up today with our speakers and address some high-risk applications and how the concerns that stem from these can be addressed via circular redesign that is based on substitution with better materials. What it means is regulatory prioritization of mitigation measures for high-risk contributors. These are agricultural plastics, fishing gear, medical devices, textiles, packaging materials. Why? Because they pose unique risks when compared to other applications for microplastics release, their exposure to soil and marine ecosystems, and also the chances of them being leaked leads to higher impacts. And how is basically the use of suitable biodegradable materials, for example, for some of these applications to promote safety are being explored, and it is argued that it can also support some of the downstream measures. So with today's webinar, we will explore the need and the feasibility of substituting conventional plastics, understand the opportunities and challenges that come with such substitution, and also discuss the cases presented by our speakers. With that, we'll start with Dr. Kirk Havens. Floor is yours.
Thank you. Let me share my screen here. Thank you, Saloni. I assume everybody can see that and can hear me. And again, I'm Kirk Havens. I'm the director of the Center for Coastal Resources Management at Virginia Institute of Marine Science, but also at the William Mary Batten School of Coastal and Marine Sciences. And thank you for the opportunity to speak today. What I'm going to do is take you on a journey that will take us through derelict fishing gear, shotgun ammunition, and bovine intestines. And so this was our journey. And you see here, if I can see my screen. And this all began with us with the issue of derelict fishing gear, and these are in particularly traps that are lost at the bottom of the ocean or in the coastal areas, and then they sit on the bottom and they can continue to capture and kill animals. That's what they're designed to. A lot of these traps are metal and plastic coated, or in some cases are simply plastic traps in the case of some fisheries. And so while this was an issue that a lot of these traps get lost, there was a real interest in how do you mitigate or reduce the impact when they are lost on the bottom, and they get lost pretty regularly. And of course, they also capture not just the target species and some of the other bycatch species, but particularly vulnerable are some of the air-breathing like reptiles like the diamondback terrapin, which is the only North American estuarine turtle. And these, of course, go in these traps and then they're lost, and they can't get up and breathe, and that's a huge mortality issue for them. And so, well, we did look at this issue while we're looking at it in Chesapeake Bay. We actually hired commercial fishers to go out and try to remove these particular traps, and the issue here was that they were using side scan sonar units so that we get a GPS location for every trap that was recovered. And the Chesapeake Bay, this is the Chesapeake Bay area here. Every red dot you see will be a derelict trap that was removed from the Chesapeake Bay. They removed over 34,000 of these. And again, they sit on the bottom, they continue to capture and kill, and they have both ecological and economic impact. And so some of them can last several years. In the particular case of blue crab traps in the Chesapeake Bay, we estimated they're capturing about 6 million crabs annually, and about half of those would die in the traps. They had an economic impact of not only the lost crabs, but the competition against the existing active fishery, which estimated over that 6-year period the program took place. Those areas where the traps were removed increased the harvest by $33 million. So it's a significant economic impact associated with these lost traps. So there was a real interest in trying to figure out a mechanism that would allow them to be disarmed when they're lost, so they wouldn't continue to capture and kill animals when they're on the bottom. And so we looked at a number of different applications, from a couple of biopolymers to different twines to even cardboard, to see whether they could be effectively used to be placed in the trap, and then over a period of time, if lost, would degrade and allow an opening to form that would allow anything that could get into the trap to escape. We ultimately settled on PHA as one that's a polymer that is actually already naturally occurs in the marine environment, and that once in the marine environment, bacteria recognize as a food and begin to metabolize. And so that one was the one that could also be molded, which is really important for this type of activity. And again, one of the interesting side parts of that is it was also that in the digestive system of submarine organisms, some fish and some crabs that had been studied, that PHA is broken down into short-chain fatty acids. And this does provide some level of immunity to some of the pathogenic bacteria that are encountered in the marine environment, which is an interesting advantage to utilizing the PHA. And this issue is not just limited to a regional area, it's a global issue. Wherever traps are actually deployed, whatever fishery is using some type of trap, there's always going to be loss, and it's estimated to be somewhere around 10 to 20% of traps that are deployed annually are lost. And so this is a huge issue globally, and so there is a real need to figure out a way to mitigate these impacts in the sense of providing some type of escape mechanism in traps when they're lost. And so besides the panel option, we also explored the issue with the Dungeness crab pots in the western part of North America. And again, this is the same type of issue where traps are lost, and then they have an impact on not only the ecological aspect of losing these crabs, but also the harvest values. And in this particular case, instead of panels, we were asked to look at another mechanism for these traps. These Dungeness traps already have an escape mechanism of a piece of cord or twine that's called a rot cord, that the idea is for it to, when it's lost, it'll eventually break and then allow the lid to be released. The problem with that is that it's variable in breakage, and the crabs need to know to push up the lid up to get out. And so that comes to be a problem. And plus, when these traps are lost, they tend to get biofouled. And so any type of biofouling could potentially cement or does cement that lid down and keeps it from being able to be pushed up. So we took a different track. There is a gate that's used at the entrance funnel, and it's held on presently with conventional plastic units that work as hinges. And so we figured, well, you can just simply replace those hinges on one of the gates or both of them with a PHA hinge. And so that over time, as they degrade, if it's lost, then these gates will just simply fall off and then open that entranceway for both ingress and egress. So we worked at this both in Alaska and in Washington State with the commercial fishers, recreational fishers actually there, and deployed these in the systems to test them out. And the final result is that it was thought that they would make a really good secondary mechanism besides the rot cord. So if the rot cord did not work, ultimately these hinges would, and it would provide an ability for the entrapped crabs and other bycatch to get out. This technology was licensed to a company now that's working on this, which is called Mardoc Marine Materials. You're welcome to go visit their website on that. But while we were working in this arena, we were approached to try to deal with this particular issue by others. And this is actually a shotgun wad. It's plastic, mostly polyethylene. And it's actually used in the shot shell, and it's used to separate the pellets from the powder, and then to protect the barrel from the steel shot when it's fired. And this, of course, is an issue because it's fired out with the pellets. And so it'll go a good 40 yards or so with the pellets, and it's really hard to either recover or retrieve. And in many cases, a lot of this is being hunted in waterfowl situations, so it's a direct impact into the marine and water environment, aquatic environments. And it's one of the common single-use plastic items found in beach cleanups. And so now when you're walking the beach and you pick one of these up, I did not know about these until I was asked to investigate. Now I know a lot about ammunition, and these are a real issue. There are billions that are discharged annually, and so one of the issues is that not only is it a piece of a plastic in the environment, but that it is mistaken as food in many cases and picked up. And in this case, this is from the Smithsonian Institute. They have a albatross chick stomach that was analyzed, and of course, yes, there was amongst a number of other pieces of plastic a shotgun wad as that was ingested in this poor chick. And again, these will, of course, break down and fragment over time. And then in our marine environment, the problem is that, of course, the smaller you get, the more things can consume you. And so as microplastics enter the marine environment, that opens up a lot of issues in the plastic pollution world. And so we were asked to investigate this, whether we could come up with a substitute for the plastic shotgun wads. And we are approached by a varied group of stakeholders, the hunters that really are concerned about adding plastic pollution to the environment, but they realize that it's really difficult for them to do anything about this since it's fired out with a gun. The environmental groups that are constantly doing beach cleanups and encountering plastic shotgun wads, the cattle farmers and the hunters that want to lease the cattle farmer property. The cattle farmers do not want plastic pollution on their farms that they're leasing to hunters, and I'll talk about that a little bit further. And interestingly enough, the cranberry bog farmers contacted us. Apparently, you get a piece of plastic wad into their harvest, and it can ruin their entire crop. And finally, there are broccoli farmers that as the broccoli comes up in these fields that have been hunted, it'll have some cases the shotgun wads in the broccoli head. And so there are a number of issues that this issue that we're interested in trying to find an appropriate substitute. So we did work on this, but before I get to that, the issue of the cattle farmers is really interesting in that cattle are indiscriminate grazers, so they'll pick up anything that's on the ground. And this includes shotgun wads, and that one looks a little bit spiky. And the issue is there's a common issue with cattle and what's called hardware disease. And this is really based on the fact that over in the years, they will pick up nails and pieces of metal fence and things like that get into their multi-chambered stomachs. And once in that stomach, the metal can get to the edge of the stomach, and as the stomach convulses, as they ruminate the grazing material, it can end up puncturing their stomach lining and go into a vital organ. So they have what's called a magnet that they actually have the cattle ingest, and this makes the metal all pull against the magnet and stay away from the stomach edge. Of course, this is not possible for plastics, so this does not work. And so farmers are really concerned about plastics getting into their cattle's intestines and causing health risks and mortality. Back to the shotgun wads, we were able to make a substitute using a PHA and compare it with some of the competitors that are well known on the market. It was very compatible and compatible to that. And so this also has been licensed to a company that is now has this working with the PHA shotgun wads, and you should visit their website as well. And then while we were working on this issue, we ended up having to deal with, we were asked to talk about the plastic control netting that's used in erosion control along highway departments. So you see them when they're making roads. It's an issue that this is laid down to try to hold back erosion until plants can colonize, but it also lasts for decades and can continue to trap stuff. While we were doing that, we were asked to also see if we could address the issue of hay bale wrap. These are these hay bales that are wrapped and put out in fields for winter fodder, and they're all wrapped with, most of them are wrapped with some type of plastic. The problem is that when this plastic freezes during the winter to the hay bale, it can fragment off and then become something that the cattle can then ingest again. And this can cause mortality in these cattle. It's really interesting that, you know, as the material moves from different chambers of a cow's stomach, at one point it needs to be about 1 to 2 millimeters to get to the next chamber. And of course, this is a real problem when you have plastics engaged in the intestines and the digestive system. And so this is another real issue. We are working with our partners at or the Virginia Tech, out in here, and the issue there is trying to develop a PHA netting that would work. And it's interesting enough that PHA breaks down in cattle's digestive system, and it will produce short-chain fatty acids, which is again a major energy source in the roles of metabolism for cattle. And so I think I am trying to make it under my 12 minutes. I'm getting pretty close. And so here are our basic recommendations are not to throw out the innovations associated with biodegradable polymers. Keep those available. With as we talk about conventional plastics, they're not the same. We need the treaty negotiators to understand the difference between truly biodegradable polymers and the conventional plastics and others. And then to try to focus on single-use plastics and plastics that are actually purposely deployed or expected to be in marine or coastal environments. And those would be our two main points for the treaty negotiators. And I think that is going to be it for me. And hopefully I made it under my 12 minutes.
Thank you so much for the very interesting presentation, and you did make it.
Hey, excellent.
And we move on to our next speaker, Yasmina. The floor is yours.
Thank you very much, and thank you for this opportunity for me to speak here today. I'm actually going to share my screen, and I hope it's in the presentation mode now, yeah? Okay, I'm just going to start. We are changing gears a little bit because I am going to cover biomedical applications of PHAs. And as you heard, they're really a great material for these single-use plastics, but they're also maybe even better for the high-end applications such as the biomedical field. So I am coming from the University of Belgrade, from the Institute of Molecular Genetics and Genetic Engineering, where I lead the group for ecobiotechnology and drug development. So just quickly, you all know what PHAs are, but they have some excellent properties, and they're called emerging material. And I think we can push that into not emerging, but really used and applied widely. So they're sustainable green ecopolymer. They are biological polyesters, and they have certain similarities to conventional plastics, and they can replace some of the conventional plastics. I'm also going to mention another biopolymer, or something as you call better materials. This is bacterial nanocellulose. Still biodegradable, biocompatible, still emerging, maybe around in the research arena even longer than the PHAs. These are polysaccharides and completely environmentally friendly, made by bacteria, and have some really nice properties that make them suitable for biomedical applications as well. So just briefly, PHAs are known for 100 years now, and slowly research has been done in order to increase the productivity and to try to tailor them for a variety of applications. And I'm really happy that I have seen some of the recent developments where we are just getting a larger volumes of these valuable biopolymers. And similarly, bacterial nanocellulose has been around, as I said, in the research arena for maybe even longer. And it is produced by several types of bacteria. This material is now making into artificial leather arena, but it has, along with the PHAs, like PHB and bacterial nanocellulose, both materials have been FDA approved because they're non-toxic, biocompatible, and have been used in biomedical applications. So let's see what we are making from them. A variety of devices and from wound healing meshes and gels and patches to some more specific applications like implants and scaffolds. They're quite good, and you can see here both from PHA on the upper part of the presentation, like in the to nanocellulose, it's in the lower part of presentation. We are making this artificial ear because I guess we are, like getting injured in that region a lot. So what is also important here, what makes the PHA really good as implants is the fact that they are biodegrading slowly. As previous speaker said, they're releasing certain hydroxy acids that are normal components of our blood, and they're allowing a regeneration of tissue. And unlike other implants with some synthetic materials that require second surgery to be removed, these just resorb into the human body. So what else is really important with the biomedical applications is that these materials have higher selling value. So biomedical, it is one of the key applications for PHA and other biodegradable materials. But we are making efforts to produce them to make them even more sustainable and to make this margin even a bit bigger. So I have been involved in two EU projects led by Technological University in Shannon, Ireland, where we are looking into getting the plastic waste, variety of plastic waste, even synthetic and biodegradable, and then making with the green mechanochemical pretreatments, we are trying to get the feedstocks suitable for microbial fermentation. And from these fermentations, we are actually obtaining both PHA and nanocellulose, and thus closing that loop of circularity. So just to show you, I'm not going to go into too much details regarding the science, but here we're just showing that post-consumer polymers, including PHA, PLA, polycaprolactone as well, and some PET, can actually be hydrolyzed. And then from these hydrolysates, we are obtaining bacterial nanocellulose in pretty good yields. So these results are quite fresh and still not published. So regarding the some work that and some cases that we have been involved in promoting and bringing PHA further into biomedical applications is our work on actually medium chain length PHAs that are more elastomeric, and even just with the solvent casting, we actually use these biopolymers as drug carriers for polyenes. Polyenes are molecules that are also produced by bacteria, and they have antifungal properties. So amphotericin B and nystatin are both polyenes and have intrinsic toxicity towards human liver. So we are actually we managed to make these kind of films, thin films, and to continuously deliver polyenes for dermatophytes. So these polyenes are usually not used topically, but in this formulation, they are more suitable to be used in the fight against the fungi. So then we were actually able to use the medium chain polyhydroxyalkanoates in the form of micelles, and also to deliver the polyenes. And we have shown that in this sort of formulation, we are reducing the toxicity of these polyenes. These are clinically used drugs. So we also looked into PHAs and more specifically medium chain PHAs to carry these bioelectronics in order to be used in diagnostics, because medium chain PHAs can withstand the high acidity of the stomach fluids. So this is something that we're looking into further developing with some companies. So what we also wanted to show you is these scaffolds, electrospan scaffolds that can be made using the PHAs, and especially in blending with the let's say another biodegradable material, PLA in this case. We made the fibers that were really good for they were supporting cell adhesion, migration, and self-organization. So this is something that can replace some other currently used scaffolds. And again, we mentioned some additives that should also be replaced. We're looking into the biopigments as well, but these biopigments produced by bacteria usually have some activity associated with them. So what we actually achieved here, we incorporated the prodigiosin, very important molecule that is showing interesting activities into the PHB covalorate. And with these materials, the biopigment represented the center for nucleation of the biopolymers, and made these films more compact with a higher crystallinity and better mechanical properties. So here we just showed that the pigment got integrated into these films nicely, and the activity of the prodigiosin was still retained in these films. And okay, I was also trying to keep within the time frame, but I just wanted to mention that within the biomedical field, regulatory compliance is something that is of a high priority. And if we were to give some key messages to UN treaty negotiators, even I'm working in the research, and I'm also always in a race for financing. I think these developing clear and unambiguous international standards to clearly demonstrate the performance of bio-based materials. It's something that is it's really of the most importance. Again, continuous stimulation of the research and development within this field is also important. At this stage, setting up demonstration projects or stimulating this networking of academic and industrial partners, it's something that it's also quite important. And of course, we would like some encouraging measures from the policy side in order to bring more attention to bio-based or better material products. And with this, I would just like to thank my team who is working very diligently on in this field, and to you for listening.
Thank you so much, Yasmina. And we move on to the next speaker. There are some questions already in the Q&A box, so if anyone wants to answer them now, or we can also pick them up live, up to you. So for the next speaker, Dr. Martin van der Say, the floor is yours.
Okay, hello, everybody. So I'm a researcher, scientist, project leader at Wageningen Food and Bio-Based Research, and I'll in the next few minutes I'll talk to you about some high-risk applications of plastics entering the environment, and that's in agriculture. So after a few brief words about where I work, I'll give you some context and some examples where we work on. So Wageningen Food and Bio-Based Research is one of is part of the Wageningen University and Research family, which is a union of the Wageningen University and a group of 9 independent research institutes that are separate entities but work closely together so that scientific breakthroughs can quickly go into practical solutions. Wageningen Food and Bio-Based Research is one of those 9 institutes, a contract research organization working for governments, NGOs, and also industrial companies on a project basis. And in these type of projects, we obviously have our different fields of expertise, and I'm working in the group of sustainable plastics technology, in which about 45 research scientists, experts work on various topics of sustainable plastics research, varying from the use of bio-based feedstocks, so looking at other formulations, also polymerization of novel monomers into materials, all the way up to various processing technologies into product development, and also looking at the end of life options such as recycling technologies and biodegradation. So my personal expertise is mainly in the field of the biodegradation research. So about bio-based plastic products for agriculture and horticulture. I think it was already mentioned earlier in the introductions also of this webinar session. Some of the high-risk applications of plastics are in agriculture, where they with the high risk of ending into the environment. Some context in Europe. I know the audience is broader, but looking from the European perspective, about 3 to 4% of all the plastics used in Europe are used for in agriculture. Doesn't seem much, but it's still 1.5 million tons per year. And obviously they're used for many reasons, where they can improve production efficiency, can be beneficial in reducing pesticides, fertilizer, and water use in farming. More recently, there's also increasing concerns on negative effects of plastic use, and also leakage of plastics into soils and waters. And there's a couple of reports by FAO, by UNEP, for the European Commission that points out these things, and and it's worthwhile to look into these concerns and what we can do about it. For example, one of these reports pictured how the agricultural plastic waste mass flow is in Europe, and quite alarming is that only about 63% of the plastics that are used in agriculture are actually reported collected after use, which means that more than a third of these plastics, the fate of these plastics is uncertain. Where do they end up in? And of this reported as collected, only a fraction is then offered actually for recycling. So recycling is not really considered as the solution for agricultural plastic waste. Remarkable is that only a really small fraction is reported to as biodegradable on land. But we should, main take-home message is that the large fraction of these plastics used in agriculture also end up as debris or soil contamination. So we need to do something about that, and obviously prevent the release into an environment and reduce the use of plastics. We also need to rethink and look at alternatives where we look at other alternative materials or other ways to deal with this. So in the waste management, recycling, whenever possible, that's a good alternative, but we also realize that it will not always be possible. And then biodegradation of materials in the environment can be a valuable second or third option, which we can divide into when we use these products in a confined environment, such as greenhouses and so on, where they need to be composted, but also when we have leakage into the open environment, we can't use industrial compostable materials. We need to take into account the biodegradation in open environments proceeds differently there. So when looking at composting in confined environments, typical applications we're looking at are products that are used in, for example, horticulture or in greenhouses, in growing of crops, where the products that are difficult to separate from the plant materials, so like tomatoes on twines and clips and netting and things like that, where it is greatly entangled with the plant material. Plant material is normally, when the crop is harvested, you have a lot of plant residues left over that are normally composted. And when you have plastics in there, that will leak into leak into the compost. And when compost is used in agriculture, then it leaks into the environment there. Obviously, main research questions when we look at alternative materials is to match the properties of these materials that are compostable with the requirements of the use cases. One of the things that we've been looking into as well in relation to this is that there's especially at least in Europe, but we know this, we hear this globally as well, that the certified compostable products, do they really fit in the industrial composting practice as they are now? And so we had a program commissioned with the Dutch waste management industry and also material producers, looking into that and to the fate of these plastics. Where do they end up? And actually, bottom line, I'll refer to, if you want to know details, there's the link in here where you can look at the public available report. But that certified compostable plastic products really degrade at a similar rate as organic waste and do not hinder this process. So that's good news if we want to use this type of products. So in the open environment, there's also challenges. We've discovered that potential sources of plastics, a lot of emphasis has been on mulch films when they are used, and they are difficult to remove after the crop harvest completely, that it leaves from 12 to 62 kilograms per hectare plastic debris, which remains then in the soil. But more recent investigations show that also formerly unconsidered applications like the coatings for controlled release fertilizers, seeds and pesticides can add another amounts in the same order of magnitude to plastics left in soil. And also unintentional introduction of plastic residues through the use of urban compost can also add to another amount of plastic into soil. So which means that we really need to look into these agricultural applications, make sure that when they end up in and leak into the soil, they are compatible with the soil and degrade there. So we've been looking at in various projects to typical products that are used there, and but of course in that sense it's important to realize that the biodegradation profile also needs to match not only the end of life phase but also the use phase. You can imagine a clip in a greenhouse will degrade differently than behave differently in the use than a mulch film or a fertilizer coating. And you need a specific approach for these types of products where programming the biodegradation is crucial. And then still keeping in mind that complete mineralization is required in the end for the total product. So in this respect, we've been targeting various type of applications and products. A typical approach is when we look at product development or experimental material development, that we make the formulations develop new formulations usually based on biodegradable materials, registered biodegradable materials, and make these formulations on the various twin screw extruders we have, and then prepare samples via sheet extrusion, which we then can expose to a controlled laboratory environment, where we can keep temperature and moisture, etc., constant in order to better compare different materials, and then follow what happens in the first stage of disintegration during this exposure, with either mechanical testing or also following the recovered materials and analyzing them further for chemical or physical properties. Obviously, the key here is that we need to translate these laboratory tests also to actual environments, which means that we have to combine lab tests and field tests in order to demonstrate a proof of concept. And in parallel, we will also need to ensure that the materials we use and additives we use are still completely biodegradable at the end. Just a few examples, starting from intrinsically biodegradable material in soil, where we were able to, with a particular additive, which I cannot disclose here, but is definitely also biodegradable, ultimately biodegradable, in which we can use with this additive, we can play around and delay the disintegration time. And as we can also see, not only visual disintegration, but also where the material maintains its mechanical properties over longer time. And we can tune this. This was observed also with increasing additive amounts or changing the additive. We can play around with that. And also the other way around, for some applications, it's in the use phase, it's needed that it disintegrates quicker than that. And also there we can use with blending technologies with other polymers or with mineral fillers to increase the degradation rate, to accelerate it, and which gives us a kind of a toolbox to program the biodegradation, the physical disintegration for the crop or application requirements, which are then subsequently put into demonstrators that are used into the field. Of course, this makes a lot possible. This was just very quick to show what has been demonstrated and what's possible. In summary, we conclude that plastic leakage from agricultural products is definitely an issue that can not only be solved by recycling. We need to take leakage into the environment seriously, and we have to match the biodegradation behavior with this end of life scenario, and making sure that not only the first step of biodegradation, the fragmentation is ensured, but also that complete mineralization is demonstrated as well. And we have demonstrated that it is possible that to tune the disintegration behavior, which is essential if you want to make products that work for specific applications. Linking this to the UN treaty negotiations, I think it's crucial when we're looking at measures to reduce and prevent plastic pollution and its impact on the environment, that we should not only focus on how to mitigate when the pollution has occurred, what can we do and how can we remove this pollution but we need to focus on prevention of pollution through the use of safe products that, if they leak into the environment, don't have safety or health issues there. And we should consider the production and the use, including also reuse and recycling technologies, and the risk of leakage in those products. Second recommendation is that definitely what we learned from the Single-Use Plastics Directive, that measures should not lead to regrettable substitutions. So we should be really aware of how definitions and measures are put in there. And a third recommendation is that we need to realize that why a lot of there are technical challenges, but most of these challenges are not relevant if they are overcome. That typically new and novel materials that could pose a solution don't make it on the market because of they can compete they cannot compete with the really low costs of the traditional materials that cause the problem. But that is because the costs of the pollution and the mitigation measures is not part of the price of these low-cost products. So we need true pricing in there as well. And with that, I will finish my words, and I understand questions will be after the last presentation. Thank you.
Thank you for the very informative presentation. Hope everyone is geared up for the Q&A. And now we have our final speaker of the day, Dr. Ipsita Roy. The floor is yours.
Thank you. I'll share my screen then, and hopefully, desktop, share, and fingers crossed it all works. Can you see my screen?
Yes.
Super. So, thank you, everybody. Last speaker is always probably, we'll definitely stick to time so we don't go beyond our stipulated time. So today I'll talk to you about PHAs, which are, as you all know, and have heard, I'm sure, from all the previous speakers, natural, sustainable polymers that are produced by bacteria. And I will speak to you mainly about biomedical applications. So it's not letting me. Let me see if that works. No, it doesn't. So sorry, I have to go back. It works that way. Beautiful. So I actually work with other polymers of bacterial origin as well. So I thought I'll mention that. These include polyhydroxyalkanoates, of course, but bacterial cellulose, gamma polyglutamic acid, and alginate are other members of the family. As you all know, polyhydroxyalkanoates are polyesters, and that's what I'll focus on today. And we divide them into two types, the short chain length and the medium chain length. Short chain length mainly being rigid, except for P4HB, which we all know is highly elastomeric, and the medium chain length, which is mainly elastomeric. And here, of course, I don't think I need to tell you again, but just for sake of completion, short chain length have monomer chain length going from C3 to C5, and medium chain length go from C6 to C16. And so this is Sheffield, where I come from, University of Sheffield, and this is our 30-liter fermenter that we have in our pilot plant, and which is where I produce my polymers normally. And here is this very typical profile that we get when we are producing the short chain length PHA in my group. As you can see, we are very, very careful, and I'm sure all of you are when you're producing the polymer, that we make sure that all parameters are very, very strictly monitored and kept under control. And this is because when we get the polymer, we want it to be exactly the same molecular weight with exactly the same monomer content, because if those two parameters change, we're actually dealing with a completely different polymer. So, for example, here are some of the properties of the poly-3-hydroxybutyrate that we have in our laboratory. And you can see that this is, I won't go through all the properties, but firstly, a very important point, if we have a solvent cast polymer versus a 3D printed, the properties change. And I will point out the Young's modulus value of this polymer, which goes to a gigapascal range, really, so 1,000 megapascal, but it is slightly higher if you 3D print it. Similarly, we also produce the medium chain length polyhydroxyalkanoates, which are the highly elastomeric ones, again, under highly controlled conditions, such that the molecular weight and the other properties, that is, the monomer content, remain exactly the same. And here too, I'm just showing you an example of a solvent cast and a 3D printed medium chain length PHA, and you can see that the properties are depend on the processing. So for example, the Young's modulus is 15 for the solvent cast and is 9 for the 3D printed structure. So unless we can make things out of these polymers, we can't really do anything with them. And of course, as you've seen in the previous talks, I'm sure we can process these polymers. So here is a review that we published, which showed how PHA-based structures can be produced. In my laboratory, we've looked at the production of porous structures, microspheres, gyrospun fibers, 3D printed structures, tubular structures, melt electrospun structures, and then melt extruded and laser ablated structures and 3D molded structures. So pretty much most typical processing methods for polymers we have explored and successfully used for PHAs. So I truly believe PHAs are new, emerging medical materials. And in my about 25 years of research with medical applications, I've used them for bone tissue engineering, cartilage, kidney tissue engineering, semi-artificial pancreas generation, skin tissue engineering, cardiac tissue engineering, controlled drug delivery, biodegradable drug-eluting stent manufacturing, biodegradable nerve conduit manufacturing, and 3D models of cancer. Recently, I've added the lung tissue engineering to this portfolio. So today, of course, it's a very short talk, and I'll only show you, or I'm only able to show you a few examples. So here is an example of bone tissue engineering. We've taken poly-3-hydroxybutyrate and created composites with inkjet, and we have very nicely, successfully 3D printed these, and without going into huge details, we see that the 3D printing fidelity increases in the presence of the inkjet additive. We see that on the right, you can see that the strut diameter decreases, the cross-sectional area of the struts decrease, and also the pore area increase. And the pores are very important in tissue engineering applications because cells which we will grow on these polymers need nutrients, need the blood supply in the body, and therefore we need the porosity for the cells to attach, grow, and obtain the nutrients. Here is an example with primary osteoblasts that we have. The osteoblasts are looking very happy and green. Red indicates dead osteoblasts. Green indicates nice live cells. And as you can see, we have more of green than red, and we've grown these osteoblasts on the P3HB scaffolds, 3D printed scaffolds, and on the inkjet scaffolds. One thing I like to point out to you, and I don't know if you can see my pointer, there are holes, obviously, or pores within the 3D printed scaffolds, but the cells seem to like the scaffolds so much, they even jump through these holes and reach or span the entire area, which I haven't quite seen with other scaffolds. We then look at mineralization using a xylanol orange assay and an alkaline phosphatase assay. And in the red indicates mineralization, which we need in a bone, as you might understand. And we see heavy mineralization both on PHB and in the presence of inkjet, but higher in the presence of inkjet. And similarly, the production of the polymer of the enzyme alkaline phosphatase, you see, increases in the blue histogram, which indicates the presence of the inkjet has helped the composite to regenerate or to produce more tissues using these osteoblast cells. I move on to another example. These are, again, 3D printed composites, but with graphene oxide. And here, again, we were able to print these 3D printed structures or grids very successfully. Again, these structures were nice and porous and therefore would help cells to attach and grow. Here again, we saw the cells that we saw in the previous scaffolds. These osteoblasts were very nicely attaching, growing, had the right spindle-shaped cells, and there were fewer red cells as compared to the green, indicating good growth on these scaffolds. I'll move over to peripheral nerve tissue engineering. As you might all imagine, nerves are very important, and peripheral nerves are very important for our movement of our fingers, our hands, etc. And if they get injured and broken, they don't repair very easily if the gap is bigger than about 3 millimeters. So what we have done is created tubular structures, which almost look like straws, but highly flexible. And then we put them across a broken nerve and seen whether the nerve regenerates. We've compared this regeneration with an autograft that is taken from the animal and placed in the gap. And obviously, we'll expect the autograft, which is a real nerve, filling the gap of a broken nerve, to work better. But here you can see an example where the nerve conduit, or that empty tubular structure, which was sutured across the broken nerve, worked almost equally well. This was in studies in mouse, and specifically in mouse or mice where the nerves are autofluorescent, so we don't have to stain them, and you can see how nicely they have regenerated. We also looked at the axon sprouting index and the disruption tracing, that is how many of the axons were disrupted and how many new axons were developing. And if we compare the conduit repair versus the autograft repair, we saw they were equally comparable and good, which is amazing because we have a real nerve and we have an empty tube, and both are working equally well in the repair. Finally, and not the least, we looked at the spinal cord of the animal and saw whether there was pain when we did all these surgeries to the patient or to the mice. And GFAP is a protein that indicates the amount of pain, and we saw equal quantities both in the conduit repair versus the autograft repair, again indicating that the conduit was working as good or as well as the real nerve, which is amazing. And this is just a quantitative graph to show the same thing. The last and not the least thing I'd like to show you is the degradation of MCL-PHA, MCL-SCL blend PHA that we found in the rat. This is, to my knowledge, the first example where an in vivo degradation of the medium chain lung PHA and the SCL-PHA. SCL-PHA has been shown, but not MCL, and this is proof that it can. Lastly, and not the least, we added VEGF, which is a endothelial growth factor, vascular endothelial growth factor, which allows vascularization, and we added that to our cultures of nerve tissue engineering, and as expected, it enhanced the nerve vascularization. And this is further example of this, which is called a CAM assay, where we take an egg, a growing egg with an embryo which develops into a chick. We put our scaffold in there and see whether it gets vascularized or not. And we actually see that there is very good vascularization. Last but not the least, I'd like to show you kidney tissue engineering. And here, again, we've tried to develop a filtration barrier using typical kidney cells, the podocytes and the endothelial cells. The red cells are not dead. They are actually autofluorescent because they have a fluorescent dye gene within them being expressed. And what we see is a wonderful cell attachment and growth. I just wanted to show you that we 3D printed this very well, and you can see that we have 3D printed 90-degree scaffolds, which is where we have exact, the grids are 90 degrees or 60 degrees and 30 degrees. And we were just checking out whether the cells have any preference, but really we see that there is not much difference, although there is some difference within the collagen production. So I'd like to conclude my talk today by saying that I truly believe that polyhydroxyalkanoates are excellent biomedical polymers. We have used them, as you see, in a variety of applications, and now we have really scaled it up as well. I'd like to thank the key scientists and my collaborators, funding bodies, and like to announce that we have now started commercial production of these medical grade polymers via a spin-out company called FAST. And we are also hiring a PDRA. If anyone in the audience wants to apply, please let me know by email. And finally, and not the least, I'd like to thank my group. I have, of course, want to finish my talk with 3 key recommendations to the UN treaty negotiators. Firstly, we need to really assess the current state of art for the production capacity of these bio-based polymers. We're trying to replace, but how much do we produce at the moment? And this production needs to be supported by the local governments so that, they are subsidized and this production is made possible in large scale. I would like to say that we need to focus on what applications are efficient, where we can use these bio-based polymers and therefore have higher impact. And again, those need supporting more as compared to let's support everything. And finally, and not the least, I find biomedical applications, these bio-based polymers, are really not being focused on. I think we need to, otherwise it'll be too late when all these fossil-based polymers will be missing. And also the main focus is perhaps only on medical packaging, and that needs to change. Okay, I stop there. Thank you for your attention, and over to our question and answer session. Thank you.
Well, thank you so much, everyone, for the presentations. There are already a lot of questions in the Q&A, and I see that you have been answering those. Thank you so much. Before we proceed, I would like to share some concerns that are directly made by stakeholders in the treaty negotiations and some discussions and deliberations very quickly, as they are called alternative plastic and plastic products, and then non-plastic substitutes, also going in a very similar direction of common concerns. But I'd like to point them out anyway, and any one of the speakers, please feel free to take some of them up and how we can address these concerns. Of course, there are concerns of financial resources and technology transfer needs for substitutes and alternatives, the potential of environmental impact, and also to avoid any regrettable substitution. The higher costs, the availability issues, and also need for comprehensive LCAs for, of course, alternatives. The discussions are in the lines of encouraging use, promotion without strict mandates for use. There are also opinions of no separate provision and treatment, same as conventional plastics, and also support for developing it further, because it is also perceived that relevant information to make mandates on this bit is not there yet. So yeah, these are some of the commonly discussed points, and I give the floor to you to comment on some of these or your suggestions on some of these. Thank you. Okay.
Maybe I can. Yeah, I can give a comment. I would say that I have been working with the PHA for past 15 years or so, and I think some of the concerns of availability and price are just being lowered recently in the maybe past year or two, because we have some big producers coming out with big volumes at the moment, and we have some developments in the upstream and downstream processing that have reduced the cost of the biotechnological production of these biopolymers. So in my opinion, these concerns are just coming really are shrinking at the moment and should not be as much of a concern. I would say some social acceptance, and some issues that can arise from just okay, why should I switch? Even if the price margin is really small, I think that should be addressed, especially in the view of the environmental impact. But for that, we need some solid data, some solid labeling systems, and I think we're on the good path. Yeah.
Thank you. Anybody else? Dr. Kirk, do you want to chip in on that?
Oh, thanks. I would just add that I think that in the sense of that the cost is coming down, and as more applications come available, then the cost will continue to come down. And I think certainly from Martin's presentation, you see that if the agricultural industry comes into play and really begins utilizing PHA in their applications, then that's a huge market that would probably again help the prices come down. In our instance, we're most interested in, issues that will handle the single-use plastics that end up in the marine environment and aquatic environments. And so that's why we look at those shotgun wads issues where, there could be incentives to have people utilize them, or simple regulations that actually say that in a public place, on public lands, you need to use a biodegradable component, and as opposed to requiring it everywhere. So I think there's opportunities to incentivize people to use these in different aspects, but I do believe the cost will ultimately be coming down pretty significantly.
But if I may chip in on that, I think reducing the cost of these novel products will take time, and it will always be really difficult to compete to similar products that are now based on the cheap bulk plastics, and it will, what I also mentioned in my presentation, I think we need a level playing field here, because these novel products, they may, even if they would be able to be made cheaper than the current polyolefins, then they probably would have already been there. The fact that they are not so much in the market is that they just have to compete with something that is cheaper. But these cheaper products have additional problems with additional costs for society, but these costs for society are not in the product price. And that's where we need a level playing field, that these costs, if you have something that contributes to pollution with negative impacts, and we need measures to do something, and those measures cost money, then that money should be somewhere in the price. I know this is something that a lot of people advocate already, and how to implement that in practice, but this is the real financial problem, I guess. And we will not be able to reach with new materials, novel materials, with specific functionalities, also not with PHA, come to the level as long as we can cheaply rip out oil out of the earth, and make the cheapest plastics from it.
Absolutely. That is very, very well put, and also discussed exactly in a similar way, that one of the hindrances
You
is
know,
that the prices on the other end are so low. So that needs to be more expensive in order to the alternatives become more affordable. Ipsita, sorry, you're
we
not
can't hear you. Mhm.
audible.
Audio is switched on. You can't hear me?
Yeah,
Yes.
now it's better.
Yeah, okay. Just wanted to say, yeah, I totally agree with what everybody is saying. And I think it's, firstly, I agree that there should be a cost to a polymer that creates environmental pollution. And what Martin was saying, that if this polymer is going to ruin our seas and create a pollution in the soil, there has to be a kind of a tax levy, a cost to that, and that will then bring the biodegradable ones more out there and useful. On the other hand, I feel that we don't always need to substitute everything that it's out there. We can perhaps start small, and we could look at applications where this polymer works better. We're not substituting the current, polyethylenes and high molecular weight stuff that's out there and doing really well and really cheap, and it'll be very difficult to replace them. Why don't we look at applications where these polymers are better, or can do things that these ones cannot? And that way we put our, we are there, and then the cost is not that important, and gradually we, get into the market, make things cheaper, and go, when the fossil fuels run out, they will have to produce some polymers using sustainable means, right? So I think we have to go slow and steady, not aim to replace everything that's out there. And in that sense, I like the biomedical area, where the cost is much less a hindrance. So if I can get a biodegradable stent that works much better than the current metallic stents, patients would want it rather than saying that
Choose.
this is expensive. They will buy it even if it's slightly more expensive. So I'm not saying biomedical devices is the way forward only, but I think we need to look at niches where we can have different functionalities which only, for example, PHAs can do or do better, and then we can really make a headway into the market.
Again, very well put. I'll pick up a question in the Q&A, and I think it's open for anyone. We're looking for an example of what is compostable plastics, and is it the same as biodegradable ones?
I can comment if you want.
Yes, please go ahead.
Compostable plastic is actually a subset of biodegradable plastic, because what we should realize is that biodegradability is a system property more than just a material property. It depends on material property, material characteristics like its chemistry and its morphology, but it also the rate and how something biodegrades is also much determined by the environment in which it degrades. And there are examples of polymers that, for example, that biodegrade really rapidly in industrial composting conditions, which are typically high solids, humid, high temperature, 58, 60 degrees C. And an example that degrades really fast there is polylactic acid. But if you do the same polylactic acid under ambient conditions in soil, its degradation rate is much lower. So you need to have some kind of discrimination between something that degrades under composting conditions and something in soil. And that's why there's different terms for that, and also different assessment methods for that. But it relates to the degradation environment. Biodegradability should always be linked to the degradation environment. It should always also be characterized in whether in what degree it is. It's full or only partially, and also a time frame. It should always be characterized by those three elements.
So just to follow up on that, and also open for anyone, there is, of course, again, concerns expressed about, because there is no global standard for biodegradability, is perceived as a disadvantage, as a challenge, and also as a hindrance in accepting materials that are biodegradable. In more global frameworks like these. Do you have any examples or suggestions of how this can be addressed?
And are you looking at me? I can, yeah. Well, I guess I think it's there's some attempts being made in legislation now, for example, in the fertilizer regulation in Europe, and also in the microplastics regulation, in which there is a kind of tiered approach for that you need to that if you have the most simple readily biodegradability assessments, if you pass those, then it's recognized it will biodegrade anywhere. And if it doesn't pass there, then but it has a typical application somewhere that you can go to typical applications where the receiving environment is simulated, and if you demonstrate biodegradation there, it should be sufficient. So that is how it is now being addressed, and I think if people understand better the thoughts behind this, it is also much more accepted.
So more performance-based, case-based
Right, yes.
criteria depending on the application and the use
Yes.
area.
We should realize that this is not only valid for plastics. I mean, this is also for paper. This is also for wood. The fact that we have oil in the earth is also because organic matter just didn't degrade because it was in an environment that wasn't suitable for degradation. We have examples of wooden ships of 1700s that are on the bottom of the sea that are still completely intact because it's in an environment that it doesn't degrade. So if natural materials that we all consider completely biodegradable do not degrade in some areas, why do we expect it from man-made materials as well? That's not realistic. But we need to make sure that the products where they end up or where they can end up, that they are part of the normal cycle, and we must prevent that they end up in environments where they do not degrade and can accumulate. But that is, yeah, not only for plastics, that is for all material, also natural materials like wood or
Very,
Yeah.
very, very well put. Yeah.
Maybe maybe we should think about I think people have accepted the scale for, like, let's say, electrical appliances of A, B, C, D, and the colors to develop something more like that now, with some labeling that you can scan, that would be so informative to say, okay, if you throw this into C, it's going to take 20 years. If you throw, like, so that people are aware, not just by some pretty green sign that, okay, if you, there is not enough information in there, because this question is much more complex. So it, like it needs more, information to be provided in that label, rather than just saying 30% biodegradable. People would not bother thinking what's behind that, or
Better labeling, more information.
more information. Yes.
Okay, thank you for that. I have a question for Dr. Kirk. You mentioned a clear distinction from conventional plastics as a key recommendation. So just would like to know, what do you see as the problem, as the bigger problem of grouping such materials with conventional plastics, and what could be the potential impacts?
I think what I was referring to in that was the concept that having regulations that would just basically ban all thermal plastics, and rather than continue looking for the possibilities for innovations associated with PHAs and others. And so I think that that's a real concern. We don't want just blanket regulations that prohibit that, and then prohibit the innovations that will be necessary to move this issue forward. And so that was what I was referring to there. And then to follow up a little bit on Martin's point, our work is associated with kind of the marine and aquatic environments. And so to his point, for some of the biodegradable components on fishing traps, it's different for what you're going to do in Alaska, where the water temperature is cold and the microbial population that's mediating this may be slower. So, but there are abilities to make those changes by just simply changing the thickness of either the panels or these hinges or whatever it is to meet the requirements that's necessary. Part of our work looks at making sure that it's not something a fisher person would have to replace constantly. You want it to be able to last a while so that it's not impacting them economically so much, so that at an end of a season or so, they would just replace these panels or hinges. As considering that a lot of these traps can last, some of these Dungeness stainless steel traps will last 15, 20 years if they're lost. And so you have to weigh and balance that, as Martin suggested.
Okay, since you have the floor, there's a question for you in the chat. talking about the how long will it take for a PHA hinge to degrade enough to open the lid?
Okay, yeah, so the hinges don't actually open the lid. They are the ones that would drop the entrance gate. And so that would then open up the ability to the entrance and exit funnels. But we were designing those to be degradable, not completely degradable, but degradable to the point where they would break and then allow these things to fall off. And that over time they would be completely degradable. But you want to make it so that they make the trap not functional, and particularly in some of these cases, for several months they would last, and then they would be able to fall apart. And that would be the key here. And then again, it really can be adjusted based on the thickness that you want and the temperatures you're working in.
Thank you. There is a question for Ipsita, and we are running out of time. So yes, Ipsita. The question is a fundamental question. What do you mean about inkjet here? And inkjet is a process, not a material.
Yeah, sorry, I should have probably said the inkjet additive that we added. So we have a company that provided us with this additive that you use for inkjet printing to the polymer, and it allows, it enhances the ability to do SLS printing, which is laser sintering. We've not been given the composition, so unfortunately I cannot give you the composition. It's definitely quite oil-based, this additive, and we added it to our scaffolds, as you saw, and got beautiful results, which we almost didn't expect. But you know, there are always things that come as surprise. We do not have the composition, but you're right. Inkjet printing is a methodology. This is used as an additive in SLS printing and in inkjet printing, and is provided to us by a company.
Thank you. And let's conclude with a very interesting question also. And it would be great if all of you can pitch in. From a policymaker's point of view, it seems difficult, of course, to promote leakage and be okay with biodegradable materials in the environment. And as the purpose was, the applications presented today are very well with environmental degradation and end-of-life scenarios. So do you foresee any possibilities to create tailored regulations that might allow the leakage of these biodegradable commodities in the environment?
Well, I'll start. I think that certainly is possible. I see already possibilities. Actually, attempts were already made to do something in the first versions of the Single-Use Plastics Directive in Europe, where there's a group of products that were regulated with various regulation, which could be extended, well, EPRs or even bans. But there's also exemptions possible, and you can make exemptions of this ban if you demonstrate that, and then have a list of criteria. And some of these criteria, which for the Single-Use Plastics Directive would have made sense, but also for the fertilizer regulation, you see these exemptions are now coming in, and also in the microplastics regulations for the use of intentionally added microplastics, that there can be exemptions of bans of products if you demonstrate that they biodegrade at sufficient rate in a relevant environment. So I think regulations like that are definitely possible.
So again, more test-based pass criteria. Show suitability.
Yes, that you need to demonstrate that it fulfills criteria, and if you demonstrate that, you get exemptions from the bans or regulatory measures.
Interesting. Anybody else? Yasmina? Any thoughts?
I think Ipsita had a hand raised.
Oh, sorry, of course, Ipsita, didn't see that. Please go ahead.
Sorry, Jasmina, if you want to say something, I can always come after you.
No, like I'm just you know, like with the policy making, and that's like I'm more of a lab rat. So
Yeah, no,
yeah, but I see the importance of this, and I see the importance not of this, like it's for this it's really important to address kids, and because this social change and attitude towards you know, materials that and I'm not talking only about PHAs, but materials and products that are better for the environment. I think they accept it quite well. And, you know, like even in shopping with my kids, I see that, you know, why don't we get this paper package? Or, you know, like they're not going for a really bright coloring anymore. And, you know, so I think there is a
yeah.
good window there to try to make this change.
Yeah, absolutely. And I think I would also add to what Martin said about the condition should be specified and the test conditions, and therefore there should be some regulation which says if this happens, it's acceptable. If this happens, this is not acceptable, rather than in general looking at microplastics, because I think microplastics also have different types. And for example, PHAs, they might degrade into oligomers. And these we know actually are food for the fish, for example. You know, this is being used as food for fish. So although I don't know whether we still consider them as microplastics, they are not toxic microplastics, which are causing, you know, marine animals to suffocate and all that. So I think the chemical nature of the degradation product also needs to be looked at. And we know that PHA degradation products are natural metabolites in living beings, like fishes, like animals, like human beings. So what we are producing is part of the fatty acid oxidation pathway. For example, I'm a biochemist and I see that. So it's not a very generic thing. It should not be, and it should be specific to conditions, to what is being produced, and therefore the banning should be really well informed, I think, and exemptions therefore have to be in place. Otherwise, we lose very important things that are not that toxic because of the banning of a generic range of plastics.
Blanket bans. Kirk, any comments?
I'll just follow up, I think, of something that Martin was hinting at, which was the idea that, you know, as we look at some type of standardization associated with this, we've been looking about biopolymers across the spectrum, so that when you're comparing PHAs, they should be compared along with lignin and cellulose. You know, those are also biopolymers, and so we want to make sure that the standardizations are, you know, comparable along those type of naturally occurring polymers.
All right, with that, we end. Thank you so much for staying with us over time, to the speakers and also the attendees. It was great hearing from you all. And yeah, I hope to see you again soon.
Thank you.
Thank you.
Bye-bye.